Polymers are large molecules composed of many small repeating units called monomers. Polymers can vary in length and can have any number of repeat units. Polymerization is the process of joining many monomers together to form the polymer. Polymers can be made by chain growth or step growth polymerization. Chain growth polymerization is where monomers individually add on to the end of the polymer chain. Only the polymer chain has the reactive group, so growth is linear. There are three main methods of chain group polymerization. Free radical polymerization is where the initiator has an unpaired valence electron, so is known as a free radical. It is formed from heating or irritation with UV light. Cationic polymerization is where the initiator is a cation, which is an ion with a positive charge. This process is particularly useful for alkenes. Anionic polymerization is where the initiator is an anion, which is an ion with a negative charge. The process does not undergo termination, so will continue until all the monomer is consumed. This means it is known as living. However, impurities such as water will react and stop polymerization. All three methods share three common steps. Initiation is where a reactive species is formed and reacts with the first monomer to generate a reactive polymer end group. Propagation is where the reactive end group reacts with further monomers leading to extension of the chain. Termination is where the reactive end group is deactivated and polymerization is stopped. Step growth polymerization doesn't rely on chain reactions. Both the monomer and the polymer chain ends have reactive groups, so all species can react together to form longer polymer chains. This means that growth is exponential. It is often required that the two monomers have complementary reactivity. Polymers can be separated into two types, natural and synthetic. Natural polymers such as DNA, amylose, and deoxyhemoglobin are made by living organisms, whereas synthetic polymers are made by chemical reactions in a lab. Plastics such as nylon and Teflon are synthetic polymers. Not all polymers are plastics, but all plastics are polymers. One of the most widely used plastics is PET. It has a wide range of uses, but is most commonly used in food packaging and single-use water bottles, due to it being strong, transparent, and lightweight. Its monomers are derived from crude oil. The diol and the dicarboxylic acid polymerize together to form the polyester. Water is also produced as a byproduct. The large aromatic ring restricts the conformational flexibility of the polymer chain and so gives PET its stiffness and strength. Its durability and chemical resistance makes it difficult to decompose in the environment. The predicted lifetime of PET ranges from 25 to 50 years. This results in it building up in landfills and littering the environment. However, in 2016, a bacterium was identified that can break down PET and use it as its sole carbon and energy source. It does this by action of two enzymes. Firstly, PETase hydrolyzes the carbon-oxygen ester bonds in the polymer chains to form the reaction intermediate. Then the second enzyme hydrolyzes the reaction intermediate into the original monomers. They can then be easily broken down by other organisms into water and carbon dioxide. However, the process is slow. It takes about 6 weeks at 30 degrees Celsius to fully degrade a thumbnail-sized piece of PET. And these enzymes are specific, so can only degrade PET. However, there is research being done to genetically alter these enzymes to enable them to degrade PET at faster rates. There is potential to one day use these enzymes to break down PET on an industrial scale. This would reduce the damage to the environment and reduce the build-up in landfills.